Hello, I'm John Hanby, and I'd like to demonstrate for you this morning the use of our field test kits. The first kit that we'll talk about will be the soil kit, and we'll demonstrate that method. And then we'll move to the water kit and show you how that test is done. The first thing that I want to address this morning is safety. This is a little bit of chemistry in the field, and as long as we do it carefully, with the proper equipment, then we don't have any safety concerns. The instructions for the kit are printed, of course, and we have both the soil instructions, which talk a little bit about the theory of this method, and then the step-by-step instructions for soil. And then we'll have the waste disposal instructions, which again brings us to the safety issue. We like to think of the safety issues as being summed up by what we call the 3 G's, and that's gloves, glasses, and gravity. That means we keep our gloves on to protect ourselves from the solvents that we use and the catalysts that we have. And of course we keep the safety glasses on. If you don't wear glasses, as I'm doing here, we have, of course, in the kit, as you'll see, safety glasses and several pairs of these safety gloves. Now the kit is itself a nice, safe way to carry this little bit of chemistry out into the field. It's a good rugged plastic case with all of the components very carefully packed into the foam cutout pieces so that you can bounce this kit across the field in the back of your pickup truck and not break anything. So let's move on to the demonstration of our soil method. This is an extraction colorimetric procedure. In other words, we're going to extract our contaminants from a soil, and then we'll take that extract and add a catalyst that causes a color to form. We'll take that color in the test tube and compare it to our soil standard photograph. In this case, it'll be gasoline in soil. Okay, the first thing you do is you remove the beaker, one of the beakers. You get 6 of these beakers in the kit, and you place it on the little electronic balance, and then you turn your balance on. And you'll note that the balance then zeroes out. Now you take your soil sample that you've collected, whether it be a scraped up sample or something from a backhoe or a core from a boring, and you quickly, so as not to lose your volatiles, you weigh out your soil into your beaker, and you weigh out 5 grams of soil. Now you have a point of diminishing returns. You don't want to take so much time weighing out your sample that you've lost your volatiles. So you try to hit it right at 5 grams of soil. And now you move quickly to your ampule, which you hold firmly down on your surface with your left hand, and then pulling up, you pop the top off of that ampule and pour the whole contents into your beaker, and you're covering your soil at this point. So you've trapped your volatiles. You've trapped, in this case, the gasoline under your solvent. Now, this is a sort of a Texas gumbo soil, but it's kind of dry, so it chops up fairly readily, and we're able to achieve a good quantitative extraction with this soil simply by stirring it around. If it's a wet, gummy clay sample, you have to sort of stir your sample around in your beaker, sort of smear it around in that solvent so that you get a good quantitative extraction. Okay, that's sufficient extraction for this sample. Now removing one of our marked test tubes, we simply pour out of the beaker up to the line. Okay, now we have, at this point, we've poured in 4.2 milliliters of our extraction solvent, which was originally 10 mils. Now we're keeping our work down low, remembering that the third G is gravity. We don't want to have this solvent up in our breathing zone, and we don't want to have this catalyst up so that it can splash in our eyes. So now we've removed the top from the catalyst vial. We simply pour that catalyst into the test tube and shake all of the catalyst out of it. Put our top nice and firmly back on the test tube and shake it up. Now we noticed that when we poured our solvent into the test tube, we had a few little pieces of our soil in there, but we're not going to let that bother us, because that should have no effect on the color other than a few little black specks, and we'll just let our eye ignore those black specks. But we're developing a fairly good color in this test tube. You get a fairly homogeneous color. That indicates, again, the amount of contamination in the soil. Okay, we had contaminated that soil with gasoline, so let's take our gasoline and soil photograph out. You give this a good 2 or 3 minutes to develop this color. In a situation where you're working with, say, a heavy fuel oil, in that instance you might want to give it another minute or so. But for gasoline, typically the color is fully developed in 3 minutes. Okay, now we have a fairly homogeneous color that has developed. This is not exactly the same hue that we have in this color photograph. This is more of a brownish, even a greenish color. This is an older gasoline sample. But the thing that we're looking for here is the intensity, or the chroma, of the color. We're looking for how heavy or dark that color is. In other words, if you were totally colorblind, you could ignore the hues, the reddish, blue, green, whatever, and just the darkness of the color is what you're looking for. So, in this instance, we appear to be about midway between the 200 and the 500 parts per million, or milligrams per kilogram. And to me, that looks a little bit, well, I'd say that's right at about 300, 350 parts per million of gasoline in soil. So we know at this point that typically we have to keep excavating. We're above, well above the 100 parts per million. Okay, now we've finished with our test. We've compared the color of the test tube to the photographic standard. And now an important part of the disposal of these reagents. We've written down what our answer is. In this case, we'll say 350 parts per million of gasoline in soil. Right away now, we want to take our waste bottle and we're going to pour the waste solvent from our test tube into the waste solvent bottle. Okay. Now, we get all of that liquid solvent out, and we see that our catalyst remains back in the bottom of the test tube. Okay. Now, we take the excess solvent that's left in our beaker, and we pour that in. But I'm reminded, actually, if you're in a situation where you have a heavily contaminated soil sample, at this point, say if our color had been off the scale, if it had been very dark or black, at this point you might want to take that extra solvent that's in the beaker, that's left in the beaker, and then using your 10 milliliter graduated cylinder, you can pour in 1 milliliter, say, of your solvent. Then, using another ampule of solvent, simply pour this ampule up to the 10 milliliter mark in your cylinder. At that point, you've obviously achieved a 10-to-1 dilution. At that point, then you can reintroduce solvent into your test tube, add another catalyst vial, and then when you shake it up and compare your test tube, then you're comparing to 10 times what you're seeing on the color chart. In other words, this chart now runs, instead of from 1 to 1,000, it would go from 10 to 10,000. Okay, that's the simple dilution procedure. But anyway, back to our disposal of the solvent. You simply pour this solvent out of the beaker or the test tube, get that into your waste solvent bottle. Okay, now when you've finished your 30 tests that you get with the test kit, you simply put your waste catalyst test tubes back into the foam, the polyurethane foam package that they came in. You put your waste solvent bottle back into the little cardboard container that it came in, and there's a package that comes with the new set of reagents that contains a CFR label that you want to be carefully, you want to be sure that is still on that package. And then you put these components back in that package, in that cardboard box, and ship those back to Hanby Environmental Lab Procedures. Okay, that's essentially the soil test. Again, it's an extraction colorimetric procedure. We've extracted a 5 gram soil sample with our 10 mil solvent ampule. We poured that extract into a test tube up to the line, and we added our catalyst, shook it up, developed the color, which we then compared to our standard. Okay, now we are going to demonstrate the water test, which is a very simple extraction procedure again. Again, we're doing extraction colorimetric analysis. We're going to extract a water sample with our solvent. In this case, we'll be using a 5 milliliter solvent amount, but we'll extract that, take the extract, add our catalyst, which is essentially the same catalyst that we use for the soil test, and compare our colors. Okay, the first thing you do, of course, is obtain your 500 milliliters of your water sample, which we have in this beaker, and now we carefully introduce that into our separatory funnel, which we use for the water test. And of course, our water kit is mainly different from our soil kit in that it does contain the apparatus, particularly the separatory funnel, the squib-shaped separatory funnel that we use for these water extractions. Okay, this is our water sample, 500 milliliters, and these are our solvent ampules for the water test. They contain 5 milliliters of our solvent. So we open these ampules up the same way that we did for the soil test. Simply hold them down and pop up the top of our ampule. Snap it off. Now we simply dump this ampule contents into our separatory funnel. You notice that the solvent immiscible with the water, of course, and it sinks down to the bottom of the separatory funnel. Okay, now we very firmly put our top back into our separatory funnel. Now you hold your separatory funnel firmly like this in both hands, and you give it an initial shake for a couple of seconds. And now we do a step here to relieve the pressure. There's a little slight pressure that builds up in this. So you simply hold this up, you shake out the little droplet of water that's up in the top of the funnel, and you let that pressure loose there. Okay, shake it up. You do this a couple of times. We're extracting our water sample now by simply breaking up that solvent into small drops with a high surface area, and they distribute throughout the water and, of course, extract the analyte, in this case gasoline again, out of the water and concentrate it into the solvent layer. Okay, we'll relieve our little pressure build up there a little bit. Now you want to give this a good 3 or 4 minute extraction. And this, of course, is a nice clean water sample that we made up with gasoline and water. Often in the real world, you'll have a much more turbid or dirty water sample that might have some particulate matter in it. In that case, you use a very different technique in the extraction, in the shaking of these separatory funnels. Whereas here, for this sample, I'm giving it a good vigorous shake. When it's a turbid water sample or cloudy water sample, you give it a very smooth swirling extraction. If you don't keep this, you'll get an emulsion layer forming if you take a water sample that's very dirty and shake it up like this. And you'll notice that there's in that sort of a sample, you would have a very heavy emulsion layer forming right here at this interface. And so again, instead of giving a 3 or 4 minute vigorous shake as we're doing here, in that situation, you'll give about a 6 or 7 minute gentle swirling extraction. That's, of course, not as efficient as breaking that solvent up into smaller droplets, so you're having to give it an extra 3 or 4 minutes in a swirling extraction. Again, that's to prevent an emulsion buildup in your layers here. Okay, for this sample, again, about a 3 or 4 minute extraction. We actually achieve somewhere in the range of about an 80% extraction of our components, of our analyte, in this case gasoline, from our water. We partition, that is, the gasoline components from the water into the solvent. But it is consistent, so we can then refer to our color chart to get those concentrations. Okay, that's our extraction. Now we place the separatory funnel back into the ring stand. We've, of course, assembled this tripod ring stand, which was contained in the kit, and that's what we use for our separatory funnel. Now, we simply remove the top from the sep funnel, take one of our test tubes. But before I get into that, here's another consideration that we want to consider. That we don't have any water, any free visible water droplets into the stem of our test tube, into the stem of our separatory funnel. And we don't in this case. If we did, you can simply take a piece of paper towel, twirl it up into a little pipe cleaner type wick, and you can dry out the stem of that separatory funnel. Okay, we want to make sure that visible water or macro amounts of water are excluded from our solvent. So we do that first by drying out the stem of our sep funnel, and now we slowly let down the solvent layer, and you can see here that the solvent layer is again at the bottom. So we let down that layer slowly until we have obtained our 4.2 milliliters, which of course is represented by the black mark at the bottom of the test tube. So we've got our 4.2 milliliter layer of extraction from our water sample. Now, let's look for water again, and this is an important consideration. We don't want to add this catalyst if we've got visible amounts of water in our test tube. That catalyst reacts very vigorously with water, so we want to exclude macro droplet amounts of water. So you can simply shake up your test tube and just visually inspect the walls of that. If there were any water in here, it would hang on the glass. Glass is sort of hydrophilic, so it holds on to water. In this instance, we have a sample that we have a noticeable amount of free water in our test tube. Now, that doesn't mean that we have a sample that we can't use. It just means that we have to take an extra step in ridding our solvent layer, or our extract, from that free water. So, a very neat way to do that is to simply take our test tube, and you can see as we showed in the close-up, you can see the water droplets along the side of the tube. So we simply transfer them from this tube to another clean, dry test tube, and the glass holds on to the excess water. Okay, so the water essentially hangs back in this test tube, and we have essentially a clear solvent layer with no water droplets visible in our new test tube. Okay, so now we can add our catalyst to that. So we take one of our catalyst vials. We can knock that down, which knocks a little of that layer of the catalyst around the top of that. And now simply add that full amount, as we did with the soil. Add the catalyst into your extract. Okay, now as before, we simply put on the top of our test tube and give it a good vigorous shake. And again, we want to give a good 3-minute development period for this Friedel-Crafts reaction to take place. Okay, we can let that sit for a minute, and while we're doing that, we can empty out our water sample into our waste containers, whatever we're using for a waste container. The cleanup of these separatory funnels in between samples can be done simply by rinsing out these separatory funnels very well. And then if there is a residue in there, or if you've had a very heavily contaminated sample, you can actually give a warm detergent wash to your separatory funnel. Then rinse it out very carefully. And one good idea that a lot of people in the field have been using is to have a small squeeze bottle of acetone available that you can then rinse your separatory funnel with. Okay. All right, we had a very heavily contaminated water sample there. And now we take our color chart, and in this case we've looked for gasoline, and so we compare to one of our gasoline pictures here. There's not really a large visual difference between regular gasoline and super unleaded gasoline, but we'll compare here and see that in fact we're somewhere between 5 and 20 parts per million. In this case, this is microliter per liter amounts. So I'm going to say that this particular gasoline and water concentration was right around 10 parts per million, or 10 microliters per liter of gasoline and water. You'll notice that these colors range quite a bit across the spectrum, but again, as in the soil test, we're looking for the intensity of the color. We're looking for how dark that color is. We don't need to pay too much attention to what actual hue is involved, whether it's red or yellow or even blue, but it's the color, the intensity, the chroma that we're looking for. Again, this sample looks to be about 10 parts per million in water. Now, that summarizes our water test. That's about everything. Let me quickly recap the safety features involved. You want to keep your gloves on. You want to keep wearing your safety glasses. You want to keep your work out of your breathing zone. Keep it down. And you want to be sure that when you've finished your tests right away, that you empty that solvent waste into the solvent bottle, which you'll then pack back into the cardboard box that these solvent bottles came in. You have your test tubes and your urethane foam. You put those back in that special box with that CFR label, and you ship that back to us. If you have any questions, please give me a call here at Help Inc. I'm John Hanby. The number is 512-847-1212. Thank you very much.